Thanks, Raquel. Now we'll have Agustín Ruiz from Fundación ACEA. Agustín Ruiz Laza is currently the Director of Research of ACEA Foundation, Catalan Institute of Applied Neurosciences. He's an MD and has a PhD in molecular and cellular biology, obtained in the University of Sevilla. His thesis was awarded the Extraordinary PhD Prize by the university. His work led to prize-winning spin-off biotechnology projects. For example, one of them won the prize for business excellence awarded by the Andalusia Council. Dr. Agustín Ruiz's work on the application of genetic technologies to medicine has translated into more than 100 papers published and several book chapters. With his work, he has helped to uncover several genetic factors related to different human diseases, including genes associated with Alzheimer's disease.
Well, good morning, and first of all, I wanted to give double thanks to the organizers for inviting me. The first is because I'm going to present a work on behalf of the Foundation, the Fundación ACEA, and other partners involved in positional cloning on different genes associated to Alzheimer's disease. And the second, more important, is for still considering me young. This is I'm very pleasant, but I will classify myself as a young man no troppo. So I am going to start speaking about a little bit about the power of GWAS to isolate or identify loci related to any disease. But very briefly, we have not more time. Because I am relatively old, I have also a little bit conflict of interest I'm showing you. And now I think I don't need to convince you that the GWAS technology is the most important technology to identify genes related to complex disorders. And in fact, during the last decade, we have isolated, for example, for the central nervous system, more than 800 loci related to different diseases in the central nervous system. And this is not casualty, because after the Human Genome Project, the post-genomic era developed a lot of technologies to isolate this kind of signals in the genome, and looking for the relationships with the different phenotypes in the central nervous system. Of course, the Alzheimer's disease is not an exception, and we have a successful decade on the cloning or isolation, or the linkage, better explain it, of different signals related to Alzheimer's disease. And I think that this information, is still under discussion about the validity of the signal we are picking up. It's still under discussion. Actually, the mechanisms behind the signals we are isolating during the last decade. But what is clear is that the GWAS technology is reinforcing several hypotheses, has been already mentioned here by Elizabeth, but using the technology, we reinforce the innate immunity, for example, in Alzheimer's disease, or any other potential entry points for therapeutical approaches completely different and unrelated to the amyloid pathology. So this is important for the field, and in the future, when we gain insight on the molecular mechanisms behind the signal we are finding out, probably we are going to obtain better drug products to treat or to combat neurodegeneration. And this is what we believe as a geneticist. Also important, I have to say that the CIBERNED teams, the Dementia Genetic Spikes Consortium, has contributed to the cloning, to the positional cloning of more than half of the genes that have been discovered for Alzheimer's disease, and that is also important. And what we are finding out is that we have about 30 genes involved in the disease discovered to date, but we are increasing sample size, looking for a very low penetrance allele with very small frequencies related to the disease. The frequency of the penetrance is not relevant to look for entry points for the disease, because what we are providing are new hypotheses to be checked in the lab after the isolation of the genes. And you can see that using the information coming from the GWAS analysis, we are reinforcing some specific hypotheses already established in Alzheimer's disease drug development, but we are also reinforcing other not well established yet. And also we are open a question regarding whether or not we have a peripheral hypothesis for Alzheimer's disease as well, because some of the genes we are isolating are expressed outside the central nervous systems. So we have to get inside in that molecular mechanism and better improve the entry points using the information we're acquiring from the genome. So just to mention, we are part of the International Genomics of Alzheimer's Consortium. This is a consortium looking for genes, and it's a meta-consortium collapsing genetic information from many different consortium, transatlantic consortium from the US and also from Europe. And we decide, we were part of the IGA paper, the most famous one, published in 2013 by Lambert et al. And we find out there are 11 genes related to the disease, and also we have a gene never reaching genome-wide significant, but are in what we say the gray zone. And we decide in cross collaboration with the University of Bonn to check this gray zone with the large data set we have available in Fundación ACE, and we try to replicate that genes, not only the well-established during the IGA paper, but also those that were in the gray zone. And the good thing was that we validate or confirmed one of these genes in the gray zone. This gene is the TRP4. I'm going to focus all my presentation, the rest of the presentation, on this novel gene isolated during the revalidation of the IGAP results we conducted in collaboration with Bonn University. And what we found is that, at least in our hands, that locus related to AD works better in the Spanish population compared to the rest of the population, but we confirmed the signal in the same direction and reached the genome-wide significant and was validated during the process. But this is only the beginning of this kind of research. It is not the very end of our research as geneticists. Now we have to look for the molecular mechanism behind of the thyroid receptor interacting protein 4 signal we have. And we start with the most obvious approach. This was to sequence the coding sequence of this gene, of this locus, because the signal was in the 11th intron of the locus, of the gene. And during the process, we find out that other people find out that friendships and stop mutation in this gene were related to very specific prenatal spinal muscular atrophy with bone fractures, a very specific phenotype. But what we found during the sequencing is that the people carrying the risk factor for AD have not any mutation, any friendships, any null mutation related to this gene. So this opened the question of which is the molecular mechanism, because it's not related to this phenotype previously reported. So we have other genes around the TRP4, and also we have other mechanisms to be explored inside the TRP4. And we have to decide, and the best way to decide is to apply novel technologies, because if we have not a coding mutation, what we have? Probably we have a non-coding mutation. And to gain insight in non-coding mutation, we gain the advantages of the whole genome sequencing coming from the ADNI dataset. And we take the information from the 15 chromosome, where the TRP4 is located, from 808 individuals from ADNI, and then we pick up all the catalog of variants associated to the TRP4 locus and specifically associated to our target allele, and isolate about 171 variants using pure genetic or pure physical information derived for the whole genome sequencing. And after that, we start annotating functionally the target variants associated to the allele on target in our study. And you can see the first take-home message from this study using the whole genome is that the linkage disequilibrium related to relatively rare variant is huge. In our case, it's more than 3 megabases. That means that our gene, our real mechanisms can be around the signal beyond 1 megabase. This is an enormous size in the genome and containing more than 50 genes I am going to show you later. But what is also clear for us is that a cluster of signals or variants isolated were very, very close in the same intron of the signal we have. So potentially the true variant is very close to the variant we find out in the original study. But we are facing with the reality of the GWAS. Based on the whole genome data, target genes for these signals are 50. At least 50 loci around the signal we find out. And we have to find out the true function related to this signal in the future. This is our challenge, a current challenge we have. The second way, if the physical data is not enough to find out the mutation, we have to apply another layer of knowledge to look for the functional variants from those that we have linked to the signal we have. And we use a set of software related to the ENCODE program developed using information from ENCODE to subselect those variants that have potentially a functional capacity to modify the expression of the genes around TRP4. And you can see that from 171 variants linked to the target SNPs, only a small fraction of them are located in enhancer, promoters, or other genetic elements related to regulation. And this was done using the FANSEC software. And also using another independent software, we found out that a single variant located 6 kilobytes downstream of our signal have the stronger score for functionality. The important thing is that this score was clearly an outlier of the score of introns when we compare the score of the mutation that are being represented in the COSMIC database. As you can see, the COSMIC is related to somatic mutation in cancer, and you have a strong selection of functional variations selected during the cancer development. As you can see, the signal we detected is a real outlier for introns compared to COSMIC mutations. We confirmed the information using another software analyzing the functionality, and again, the same mutation was exactly the top signal or all the variant linked to the target SNP we are researching. So in conclusion, we found out using the whole genome sequencing a variant which is in complete linkage disequilibrium with the target SNP that we detected during the GWAS stage. This variant is located only 6 kilobases downstream of the GWAS signal. This variant is intronic as well, and is located in an enhancer within the TRP locus. And this enhancer is binding with very different but important transcription factor related to the stability of the expression of the central nervous system, and also that 3 independent software have proven functionality for this variant. But this is not enough, because we have to prove that the variant located is actually functional. And now we are conducting epigenetic studies. We are back into the bench looking for evidences about methylation in this locus. We are looking for information in AQTL studies, has been published. We have conducted TRP4 expression analysis in the brain and other tissues, and we are conducting functional study looking for crosstalk of the TRP4 with different hypotheses around Alzheimer's disease. We are exploring, of course, the enhancer activity with and without the mutation, and of course, we are looking for an integrative analysis, looking for more clues about the true signal behind the TRP4 locus. To finish, I want to thank to my team, of course, in the foundation, but also many people contributing to this project, especially to the University of Bonn. We are working very tight with them, trying to characterize this mutation, and also the Catherine Bayer team in German Central Puyol Hospital. They are working on the brain immunohistochemistry. Also, the Centro Andaluz de Biología de Desarrollo, Manuel Muñoz, is developing the technologies to test TRIP4 in the context of C. elegans models. And, of course, the people from the Centro Andaluz, the Studio of Informaticos, they are conducting the integrative analysis. And all the consortium will participate in. Thank you very much.